showcase exhibition hall. This is the room right now.
You can't smell that. Really? This one smells really nice. I like that one. That one smells.
Hey everybody.
That's quite extreme. So we crushed 50.
Never reach maximum velocity. So a maximum velocity for an enzyme can be considered to be more of a theoretical concept. We can calculate it, and certainly you'll be calculating it, and it is used as a measure of an enzyme, but in practicality it's not something that you can actually achieve with an enzyme, because enzymes as proteins can degrade over time, and as a result of that their activity is going to be lost. There's competition, so substrate comes in and product is coming out, there's going to be a delay, so there's going to be a point where having more substrate doesn't mean that more product is going to be generated. That's really at the core of what we're trying to understand. And so when you look at this and you have different levels of substrate, so increasing substrate on the amount of product generated in a fixed period of time, that as you increase goes higher and higher, but you notice that it starts to plateau, that these are not linear as you might expect, which is what the dotted line is showing, but it goes up and then it starts to slow down. So we're reaching that maximum independent of the different substrate concentrations. So there is an equation that you need to know, and it's this one here, the Michaelis-Menten equation. This is really critical because this equation allows us to calculate and determine different parameters, and really the two main ones are going to be Vmax and Km, that are important for enzyme activity and for reporting enzyme activity. And these are two of the parameters that you're going to be calculating in your enzyme kinetics practical, which is much later this semester, but you'll be using this idea, and some of the images that you'll have in your notes, you are going to be replicating some of those graphs and those processes. So here we see V0 relates to the enzyme's initial reaction velocity. So it's just really, if we're starting at the beginning, it's going to be 0 because there's no conversion of substrate to product. Vmax is that theoretical maximum velocity. And so that was the dotted line that we saw. This is where it reaches that point. And notice that it says theoretical. S is the substrate concentration that we're using, so how much of the substrate has been added, and so micromolar typically, and Km then relates to Vmax, and these two are interconnected in the sense that Km is the amount of substrate that gives you 50% of Vmax. Now we use 50% because we can't achieve 100%, so 50% we can achieve it, and so therefore it's a much easier thing to measure. And so this diagram again is showing you that here is substrate, here is reaction velocity. As we're going up and up and up, this is eventually going to meet this point, but when we get to half of that reaction velocity, so this is midway between these data points, we have Km. Now one of the problems with this diagram is that this line has not reached Vmax, which is up here, so really half of it should be where it is, half between this line and this line here, not between this point and this point. So if we don't know what this is, how do we calculate Vmax? Sort of stuck in a bit of a chicken and egg syndrome here. So the idea is that we need to manipulate the data, we need to change the way the data is presented, and this will allow us to calculate both with a little bit of mathematical sort of trickery. And it might look a little bit complicated at the beginning, but it's actually fairly simple. And so what we need to do is you'll be generating a double reciprocal plot or a Lineweaver-Burk plot. And this is what we do to manipulate the data so we can calculate both Km and Vmax, even though Vmax is not achieved in that graph. That's what this graph is showing you. We have on the horizontal axis or the x-axis, we've got substrate. Remember, we've got 0 in the middle, so this means that on the right-hand side we have the amount of substrate we have added. But similarly, we have a negative axis. That's probably one thing that's going to confuse you at first. You think, well, how can you have negative substrate? How can you have minus substrate? This is used just as a calculation point, and we're going to extrapolate past to intersect here. You can't obviously have negative substrate and then have activity of the enzyme. So it's there purely from a calculation perspective. The vertical line or the y-axis is your velocity. And you'll notice that these are 1 over, so 1 over v, 1 over s. So these are referred to as the reciprocal. So when you take 1 divided by something, you're taking the reciprocal. So everything you see on the right-hand side, this slope of this line, you can calculate that. You can have substrate concentrations, and you can measure the velocity, and you can plot that, and you'll get a straight line, depending on the quality of your data. And so when you join these together, you get a straight line, and then all that we're doing in this example here is that we extend this line, we keep the slope the same, we extend until we intercept back here. And it is this intercept, the vertical intercept, which is our 1 over Vmax, and this one here, which is called minus 1 over Km. Now, what does that mean? This one here is probably the simplest one to understand, this vertical axis. Because this is velocity, where this interacts is 1 over Vmax, because it's a reciprocal. But when we then take that number and then divide it back into 1, we go back one step. So we go back to actually calculating what the Vmax is. So it allows us to work out Vmax, which we couldn't do previously. This value back here is then the minus 1 over Km. Now, ignore minus 1 or minus. It's only minus because it's in the negative concentration range here, which we said you can't have. So it's 1 over Km. So again, it's the same idea. If this is substrate, then this is telling you how much substrate is equivalent or is required to give you 50% of Vmax, this value here. Allows you to calculate those two. Now, this is all going to be very much theoretical to you at this point because you haven't done the practical. You haven't played with these numbers and gone through the process. But what I suggest you do and highlight in your notes is come back to this with the enzyme kinetics practical, because we will be doing this. You're going to be drawing this graph, and one that I'll show you later, twice. Okay? And similarly to one of the other graphs that we've seen already. Here we can see the differences in Km values. So you can see that some enzymes, like chymotrypsin, Km is 5,000 micromolar, so 5 millimolar, quite high. which we've got lysozyme, is only 6 micromolar. So there's quite a variety depending on the enzyme. And it really depends on the nature of the substrate, the amount there, the reaction, whether it needs to be a fast reaction or a slow reaction, etc., as to what is occurring. But you can compare based on those when the reactions are going to occur. And then if you have a series of enzymes that are in sequence, and you know what the Km is, it then really determines, well, if one has a Km of 5,000, what's the Km of the next one? It's going to be dictated by the previous enzyme, because that one feeds into the next reaction. And so you can sort of have a better understanding of the entire cascade as it is starting to develop. Now, notice here, when substrate is low compared to the Km, so if you want 50 micromolar for penicillinase, but you've only got 5 micromolar, the enzyme is going to be very, very sensitive to changes because you're starting to move, but really it's not going to give you much of a difference. So if you go from 5 to 10, yes, it's a doubling, but we're still 5-fold away from the Km. So we're going to get a small increase, we're not going to get a massive increase. When you have the substrate at Km, so that 50 micromolar, that's when you're going to have activity. And it's also really where if you go higher, well, you're not really going to see a lot more because we're starting to then go into the plateau phase of the enzyme. You might double it, but remember, you're not doubling it to 100% because you're starting to plateau. There's these issues with the enzymes actually functioning with the substrate, the degradation of the activity of the enzyme over time. So what you see here, cells typically operate with a Km which matches the physiological level of the substrate. So these values here are what you would predict within the cell. And that is also the concentration you would predict in the subsequent enzyme, the subsequent process that we can go across. And then there was this simple calculation here just saying, well, if I've got 1 micromolar of carbonic anhydrase, so 10 to the minus 6 molar, it forms 0.6 mole of hydrogen carbonate per second. When it's saturated, so at that maximum, what's the reaction? How fast is it occurring? And so you just divide one by the other, so 6 by 10 to the 5. So every reaction occurs 1.7 microseconds. Incredibly fast, right? It's a massive turnover at this particular rate. And so we can calculate what is the speed of a different enzyme. And you see here the turnover number, carbonic anhydrase, which is really very important for CO2, as we're breathing it in and out. You can see 600,000 reactions per second, and others very, very slow. And it really is dependent on the nature of the reaction. If you have a reaction that needs to take a substrate and move it out of the body and convert it into something before it becomes toxic, you must react with it really quickly. If you don't, it's going to become toxic or it's going to have side effects. In other cases, like the lysozyme, well, if it turns over and just breaks something down and degrades it, it probably doesn't need to be super fast. It just needs to be able to do so in order to break things down. But you can see there's a whole spectrum here of different activities. And different orders of magnitude as well. Now, in terms of enzymes, we typically think, okay, well, an enzyme does one thing, which it does, and so you can have either a sequential reaction, so enzyme converts into one product, that intermediate then gives you rise to a second product, and then you have your final output, or you can have double sequential. And this one here, this is something that we will see in or double displacement, I should say, in the metabolism type lectures, where basically 2 reactions are occurring. You have 2 substrates coming in, one is acted on, generates a second, then you generate a third, and then you generate the fourth one. So you have 2 displacement. They're also referred to as a coupled reaction because the 2 are sort of working in tandem. I put this here, not for you to worry about what is occurring and what is happening. It's really just to give you the idea that you can have one enzyme acting on a substrate to give a product, or you can have multiples coming together and they fuse together. This is important when we come to the metabolism content, because we will see this, aspartate, oxaloacetate, alpha-ketoglutarate, and glutamate. We're going to see this in the citric acid cycle. Okay? And you'll see these coming together and generating this product. So it's important you have an understanding of what's occurring. So here, double displacement, first substrate, which is aspartate, binds to the enzyme, first reaction leads to a product, this leaves, and the second substrate comes in, and so forth. So you can see, it's like they're exchanging position. Okay, now, allosteric. This is where it gets a little bit more complicated. The allosteric enzymes, what we're talking about here, enzymes follow Michaelis-Menten, that's fine, which means that as substrate goes up, the product goes up. We have to be able to regulate the activity of an enzyme, and one way to regulate the activity is through the products that are generated. So remember, a substrate that binds to an enzyme gives rise to a product. That substrate could be the product of the previous enzyme, or if it's right at the very beginning of an assay or beginning of a reaction, that substrate is the molecule that gets acted upon to generate a product. And so if we look at this diagram here, A, B, C, D, E, and F are just enzymes, or processes, substrates. E1, 2, 3, and 4 are the enzymes. So you can see here, you've got this substrate being converted into a product. This product now becomes the substrate for the next enzyme, and so on. And so these are like dominoes. If you look at the blue line, this final product of this synthetic pathway comes back and inhibits the first enzyme. This first enzyme is referred to as the rate-limiting step, because this one here controls the activity of all of the enzymes that are downstream. And so because of that, this product is able to turn off the entire cascade. And this is a way of limiting overproduction. Once product F reaches its threshold within the cell, you want to stop it being fed through and being generated. And the best way to do that is to stop right at the beginning. If we stopped further down, or other places, these are still feeding in, these other molecules are starting to go up. So we want to stop right at the very beginning and you stop the entire cascade. This second diagram is showing you the same thing. So if you're going to keep going, these go down to different products. But what you can now see is that we've actually got 2 pathways that are sort of fusing or coming together. And before we said that F came back and inhibited, which is true, but F can also go on and interact with a different enzyme and now stimulate the activity. Because this product might also be involved or be the substrate for a different pathway. And so when this comes in, it needs to be acted upon and therefore has to stimulate the activity of another enzyme. So really the key point here is that we can inhibit or we can stimulate the activity of enzymes. And we can do this through the products that are generated by a pathway or a different pathway. And in turn, this controls the flow of metabolites within the cell. These graphs are here just to try and demonstrate, so we've got substrate versus reaction. You can see the standard approach, Michaelis-Menten, and here with the substrate concentration with an allosteric enzyme, it shifts it to the right. So you have to have a higher concentration before things occur, because we've got that dampening effect that is occurring. Now, this is where it can get a little bit confusing. What we're talking about here is the way that biochemists have tried to explain, how does this occur? What is the model? How does an enzyme physically work? And they've come up with an idea to say, well, look, this is one option that might occur. And they use two terms. So enzymes can exist in two different configurations or states. R for relaxed or T for tense. Think of it a bit like on and off. Okay? That's probably a bit of a simplification, but it's a good starting point. What we have is that if you have just the enzyme, you've got a mixture between the relaxed and the tense, so the on and the off. But if there's no substrate, there's not going to be any product because there's nothing for it to act on. And these two states can interchange because of energy, input, etc. that comes from the environment. What we want to see is that the active sites of the enzyme have to all be in the same state. So the tense, the relaxed, but not in a hybrid state. And that's where we start to get the activity. And of course, the substrate is going to want to bind to the relaxed, which remember was the on state, much more preferentially than in the tense state or the off state. Remember, what we're talking about here is the substrate binding to the active site. If the active site is in a configuration where it is slightly modified, so it's in that tense form, substrate's not going to want to come in. As it starts to shift into more of an open form, substrate is going to come in. And we'll get conversion when all of the active sites are occupied. And so in the diagram below, this is showing you the enzyme just as 4 squares when it's in the tense. Here we can see the enzyme in the relaxed form. So you can see most of it is in the off state. Then we start to see that we've got enzyme with 1 substrate being bound, 2 being bound, 3 or 4 being bound. And it is in this transition that we start to get more of that catalysis that is occurring, more of that shift. Now you might be thinking, well, this is all well and good. It's very confusing. Why do I need to know this? It's important from the concept of trying to understand how an enzyme acts on a substrate. So this is more of a theoretical principle. Biochemists trying to figure out what is going on, why is this process occurring, and what are these processes that are occurring. From my perspective, I deliver it to you just to give you an idea. This is where we're thinking of how things are going. We're not going to spend a huge amount of time delving into the depths of this because it is so complicated, and I really think you are much better served by understanding the fundamentals of enzymes, kinetics, substrate, etc. Those concepts. This is a much deeper level of understanding that we don't need. Okay, and I'll admit, I don't understand this myself 100% because it is such a niche area. And really, it's beyond what we need to know at this stage. We need to understand what is occurring at a whole biochemistry level before going into this detail. How it actually works in this context, I don't think it's super critical. If you understand substrate, product, and regulation, that's a much more important aspect of cellular function, right? Going into the detail of, well, is it one substrate or one subunit binding, etc., doesn't really matter too much. It's more the big picture. So again, it's here just to give you that overall idea of what biochemists are thinking is occurring. And you can see that as things change, allosteric enzymes will have a shift, and this shift allows for benefits in terms of either Michaelis-Menten or different changes in activity. So you see these shifts in these enzymes, and that's what these diagrams are trying to show you, that at different substrate levels, you have different amounts of velocity occurring, so lower here and much higher here. Now, this is a good example, though, of regulatory action. So aspartate transcarbamylase. Some of these enzymes you will see later, and this is why this one is put in here. So aspartate, as an amino acid, is a substrate. And so in this case, it's a substrate for this enzyme. You can see that the black line is the standard amount, standard activity, so the rate of formation. If we add ATP, we can see that we get a shift to the left. If we reduce or we change the substrate, so CTP, we see that there is a shifting to the right, so we get a reduction. So ATP is an allosteric activator of the enzyme. And we can see that because the line is shifted to the left. We need less substrate, so aspartate in this case, for formation, so we're stimulating the activity. And similarly, CTP, so cytidine triphosphate, stabilizes the tense state, making it harder to bind substrate. So it's like we're inhibiting. By stabilizing the tense state and not the relaxed state, we're not going to get catalysis. So think of it like an inhibitor, we're blocking the reaction. And you can see that we've got now a substantial shift to the right. And look at the numbers. 2 millimolar ATP, we're getting a small shift, but it's there, but only 0.4 of CTP, so much, much less. But notice the massive shift to the right. So small amounts can have a big effect on the enzyme, on the activity of the enzyme. And by dialing in those subtle changes, we can turn the enzyme on, we can turn it off. And that's very much similar to what we saw here. The ability to inhibit and the ability to stimulate depending on the presence or absence of that molecule. Okay, now I think it was important to spend time going through that, but can I just double check with you, either in the chat or with thumbs up, does that give you a general overview, a general understanding of the importance of those key points and the areas that I want to focus on? Thanks, Eloise. If it's not clear, please let me know. Thanks, Anna, Aaron, Nathan, Mimi. Okay, fantastic. It's up. It is stimulating the activity. We've already covered this, but I think it's good to go through it again to highlight. give the Michaelis-Menten equation, define all the terms. The equation can be arranged or rearranged to give the equation for a straight line, which we saw. When this is done, values can be calculated by the respective x and y intercepts. So that's really what I focused on before. So here is the Michaelis-Menten equation. You need to know it. Okay, you need to know what it is. So Vmax, substrate divided by substrate plus Km. If you change substrate concentrations, that feeds into the equation, and that feeds into one of the questions that we've got below. Okay, so think about how that is going to impact and affect the reaction. We spoke about those different components. Remember, V naught, it's either V naught or V. I will typically just call it V because it's our starting velocity, is Vmax times our substrate divided by substrate and Km. So Vmax is that theoretical maximum. Substrate is how much substrate we have. So if it's 0, you've got no activity. And Km is how much substrate you need for that 50% process, or 50% to be achieved. And 1 over Vmax, or minus 1 over Vmax, was the vertical intercept, and minus 1 over Km was the Km of the enzyme, the amount of substrate that you need. And as I mentioned, you're going to do this in prac class. Now looking at this, you might be thinking, wow, that's very complicated. And yes, I can appreciate that it looks complicated at the moment, but when you get your data, you'll see it's actually very simple to calculate. You'll generate a straight line, you'll be able to work this out very, very easily. So it's a nice way of getting these data points and getting these values, and then that allows you to compare between one enzyme versus another, and whether an enzyme is going to work. Have I got enough substrate? If the Km is 5 millimolar and I've got 1 micromolar, well, how much activity am I expecting? If I give it 1 molar, is that excessive? And in both of those answers, the answer is going to be, well, it's not going to work, and yes, it's clearly excessive, and it wouldn't work within a physiological system. So it helps you to understand where things are going. Okay, so what do people think then for this one? Enzyme that displays Michaelis-Menten kinetics the most. If it's assayed at a substrate concentration that's 4 times Km, what's the predicted reaction velocity? How would you go about answering this question? Probably a difficult question, to be honest, but it's one that we will work through. Okay. So let's have a look here. You need to go back to the Michaelis-Menten equation. So here it is, Vmax, substrate Km. So substrate concentration is 4 times Km, so S is 4 Km. That's how we would write it. And we substitute this into the equation. So Vmax times 4 Km, Km plus 4 Km. Now we've got the same thing at the bottom, so we simplify. So it's 4 over 5. So therefore, it's going to be 0.8 Vmax, so 80% of the maximum velocity. Notice, you're not going to reach, okay? Those values are never going to reach because you're always adding to the next one. That bottom or that denominator is playing a role in what is occurring. That's essentially how we use this equation. You plug in those values and you're able to calculate what that percentage would be. So this is a good example that we've got 4 times Km. So if you're saying 50, well, that's 200 micromolar. You think, oh, I'm going to get close. Well, I'm getting to 80%, but I'm still 20% away, aren't I? And remember, you might go even higher, but I can't get higher. I'm not going to get to that 100% because it's that theoretical value, is that value that you're not going to be able to achieve. But this allows you to calculate that percentage. Okay, this one here is similar to what we had in the other diagram. 8 by 10 to the minus 6, so 8 micromolar solution of acetylcholine esterase gives you 0.2 molar of choline per second. Remember, it was just one divided by the other. This gives us 25,000 reactions per second. So, slower than the example in the notes, 4 microseconds, but still incredibly fast. 25,000 reactions per second. What does this tell you about the reaction? It tells you this must be a really important one because the cell is metabolizing substrate into a product really, really quickly, 25,000. Whereas we saw before that there are examples of half a reaction per second. Okay, so 2 times a second you've got something going on, so fairly slowly. Okay, now this is one that you will be able to answer in terms of inhibition. How do we look at, or what are the changes in Km and Vmax with different types of inhibitors? This sort of follows on to the next lecture, but you should be able to bring the two together. What do you think? Are you all just totally confused? Give me a thumbs up if you're confused. Thanks, Anna. Okay. Remember, we've got 2 different types of inhibitors. We've got competitive and non-competitive inhibitors. And I'll go into this in more detail in the next part of the tutorial. But remember, think of competition. A competitive inhibitor is the inhibitor and the substrate are competing. Thanks, Ken. Feedback inhibition. That's part of what we're doing, but in this context, it's slightly different. Now, if we think that an enzyme, an inhibitor, and a substrate are competing, as it says there, they're competing for the active site. They're competing to want to bind. Now, for the inhibitor to be able to compete with the substrate, must it look the same as the substrate, or should it be a different shape as part of the competitive inhibitor?
It just needs to resemble the substrate, so it can be partially, as long as it blocks the substrate from binding.
Perfect, thank you. So it needs to have some similarity in shape, because otherwise it's not going to fit, okay? So it affects substrate concentrations. So if you have an inhibitor that has the same shape, it's able to compete. So the ability to compete, if you imagine that at a ratio, if you've got one molecule of inhibitor and one molecule of substrate, there's a 50-50 chance of the enzyme taking one over the other. As we start to increase the concentration of inhibitor, there is more chance that the inhibitor is going to bind to the enzyme, and that conversion of substrate to product is going to decrease. The velocity is going to decrease because less substrate is taken up. And if we measure, remember we've been talking about how many reactions per second. If 50% of those reactions are on an inhibitor, there is no product. So from 25,000 reactions, we go down to 12,500 reactions per second. And as we start to go higher and higher, we might get down to only a couple hundred or zero if we're able to completely block that enzyme. So increasing the level of inhibitor can outcompete the substrate. And the only way we can correct this process is that we have to increase the amount of substrate. So in my example, I said that we had a 1-to-1 ratio, and I said that if we increase the amount of inhibitor, the inhibitor has more opportunity. So we're going this way. Now, one way to do that, obviously to correct that, is then just to increase the amount of substrate. So there's more substrate molecules that are present that are able to compete. The cell might be able to do that, but also what you will find in a biological system is that inhibitors have a half-life. They don't just exist forever. So you know yourself, if you've got a headache and you take an aspirin, you take that, and then it says after 4 to 8 hours, if pain persists, take another one. Why? Because the enzyme, or that inhibitor, is acted upon by an enzyme that degrades the aspirin. And so those inhibitory effects are lost, and we start to have more substrate being produced. So the inhibition is outcompeted. So you need more substrate to reach the same level of activity. It's the only way we can counterbalance between the two. Now, let me go back to you all. So either chat or in the thumbs up. Did that make sense? Do you understand how a competitive inhibitor is working? Thanks, Eloise. Madison, Anna. Now, I'm not sure if that's Megs or Megs, sorry, but thank you. Emily. Okay, fantastic. So remember, just think this is competitive. So non-competitive has to be completely different, okay? It does not affect the active site. Now, if we have an enzyme, we have the active site in one location and we've got everything else. If it's not affecting the active site, the non-competitive inhibitor must be somewhere else. It must be anywhere else that is not the active site, and that's what this is referring to. It's at an allosteric site, and we've spoken about allosteric sites now on a number of occasions. And what this does is that by binding to a different portion of the enzyme, it can change the shape of that enzyme, and in doing so, it affects the shape of the active site. Now, I had an example the other day where the student came to see me, and I used my key, and I said, well, you imagine a key, you've got the handle, and you've got the ridges of the key. The ridges of the key are the important parts of the key. That's the active site. That's where the action takes place. The handle, but no one cares about the handle. As long as you can hold the key, it has no impact on that particular conformation. Now, if we use that same analogy, we imagine that that is an enzyme. By having a molecule that binds to the handle, that portion, it changes the shape of the enzyme, and maybe that zigzag, which is the key, bends. And by bending, the substrate can no longer enter. It's changed its shape. So if you imagine if you took your key and you hit it with a hammer, right? Those things are going to bend. They probably won't fit into the lock, and if they do, they're not going to work as you would expect. That's an external modification that has changed the active site. And so that's what's occurring here. Effect is independent of substrate because it's not substrate going in, it's just we're stopping the substrate from entering because of a distant modification somewhere else. So Vmax now becomes altered. We drop our Vmax, but the amount of substrate that is required stays the same because it's not to do with a competitor, it's just a distant activity that is occurring. And so it's due to those major changes in the binding site that we've seen. So again, let me go back to you all. Does that one now distinguish from competitive? Does that now make sense on why the activity? Thanks, Erin. Eloise?
Thanks, Maurizio.
Yep.
I just had a question. Is it wrong to say that there's multiple active sites that are combined, an inhibitor and an enzyme combined at the same time, or do you have to specify that it's an allosteric site and an active site?
Oh, look, when we talk about the tense and relate, that's a really good question. They talk about multiple active sites because you might have multiple domains, so you could have more. I think for what we're going through, let's just keep it simple and say you've got one enzyme, that enzyme has got one active site for one substrate to come in. And that makes it simple. You've got binding or not binding, you've got a one-to-one interaction. If you had multiple, it would be the same. And remember, the inhibitor would then be able to compete with the others. But I think let's just keep it simple, just say one-to-one. If we've got then the non-competitive and it's at a different location, so if you imagine my hand, this is probably good, another example as well. Imagine my fingertips being the active site. When the substrate comes in, and let's say it's the pen, it can now bind. If I was to remove my thumb, I can't really hold it very effectively. And by removing my thumb, it's because I've changed the shape of my hand. Don't have a hybrid between the two. And substrate and okay, excellent, thank you for, and you can see here the inhibitor has the same shape, not exactly the same, because if it was the same then it would be the substrate, but it comes in and is able to bind, and in doing so it is occluding or blocking that substrate from coming in. And so what we can see here is that as we start to have no inhibitor, we have a normal curve, substrate goes up, we reach a plateau and we're getting close to 100%, but as we start to increase the amount of inhibitor, notice that we start to flatten. So let's say it's 1 micromolar, there's no units here, but whatever it is, 1 millimolar, 5 millimolar, 10 millimolar, we start to dampen it, and as we get higher and higher, we're going to get more and more inhibition. Why is it not 100%? Because in some instances, the substrate is going to be able to bind even though there is an inhibitor present. It's really just a ratio. Now we can explain this. What is the change? You can see that the competitive inhibitor has increased Kn. Now notice, why is it increased? Because the Kn intercept has gone closer to the right. Remember, this is a reciprocal, so it is an inverse value. If it went to the left, it's actually getting smaller. Okay? Now typically we go from 0, we go up, but remember the opposite of up is down. So it means the concentration is going down, so we're getting smaller and smaller. So therefore, going to the right, concentration is going up, Vmax stays the same, and Km is increasing. Now, of course, this is perfect data, but you can actually demonstrate this, and you will demonstrate this in the prac class. Going back to the previous comment about different shapes, we can see here methotrexate, we've got other compounds that interact. Use that as acids, where we had the left and the right, sort of the processing. They're part of the pathway, but I'm not expecting you to memorize biochemical pathways. Okay, they're lower in the order of understanding and consuming process. Ribose is a sort of emoji or symbol. It's not at a distal site, it's somewhere near the active site. This is rate. Excellent, thank you. Again, and the reaction. Are there any other questions about this content? Let me know in the chat, or just feel free to unmute yourselves. Or let me know if it's all good for your thumbs up. Thanks, Emily, Madison. and Anna. Excellent. Thank you for responding. So let me just close this one, and now I'll open up and go to 7. Because that one doesn't, we didn't have the tutorial questions, but what we're going to show here now is those inhibitors and those basic processes. So you can see here, we've got that example of competitive and non-competitive. So as I was saying to you before, here is the enzyme in yellow. Here we can see this indentation being an allosteric control point. And you see here the inhibitor has the same shape. Not exactly the same, because if it wasn't the same, then it would be the substrate. But it comes in and is able to bind, and in doing so, it is equivalent of blocking that substrate from coming in. And so what we can see here, what we can see here is that as we start to have no inhibitor, we have a normal curve, substrate goes up, we reach a plateau, we're getting close to 100%, but as we start to increase the amount of inhibitor, notice that we start to flatten. So let's say it's 1 micromolar, there's no units here, but whatever it is, 1 something, 1 millimolar, 5 millimolar, 10 millimolar, we start to dampen it, and as we get to higher and higher, we're going to get more and more inhibition. Why is it not 100%? Because in some instances, the substrate is going to be able to bind, even though there is an inhibitor present. It's really just between the ratio. Now we can explain this. What is the change? We can see that the competitive inhibitor has increased Kn. Now notice, why is it increased? Because the Kn intercept has gone closer to the right. Remember, this is a reciprocal, so it is an inverse of the name. I'm highlighting role of the glycolytic pathway. This is going to be another point that is important. Kinase, P of K or PK as well, and those control points and how this will occur. Glucose is converted into 2 molecules of glyceraldehyde 3-phosphate, as we've got here, to the reaction. That is the thing to be focusing on. When we go through and we say, well, this enzyme does this, and this one does this, and this is sort of the processing, they're part of the pathway, but I'm not expecting you to memorize biochemical pathways. Okay, they're lower in the order of understanding and remembering that is required. And you'll see this in bold, you'll see this being highlighted in my recordings, and you'll see this being highlighted in the focus of the tutorial questions. Okay, that is vastly important, because if you don't, you're going to get overwhelmed and you look at it and think, oh, he wants us to remember all of this stuff. No, I don't. Okay, the other point to remember, these lecture notes are telling you a story or trying to explain concepts. If I just said to you, 1, 2, 3,
welcome to the next lecture, lecture 8. And in this series, we're now going to move into talk about carbohydrates and importantly the metabolism. So really for this topic, we're just going to talk about carbohydrates and their important role for energy, as well as some of the characteristics of carbohydrates. And so you'll see here we'll talk about describing biological roles, the importance of carbohydrates, understanding the different forms that glucose can exist in, and this is going to be the principal carbohydrate that we will focus on. And we'll also look at how glucose molecules are joined together. This is going to be important when we look at it from the perspective of storage. So space for the questions and reference to your lecture notes in the textbook. So we know that carbohydrates are one of the 4 major classes of biomolecules, and we know that the others are proteins, lipids, and nucleic acids. But some of the characteristics of carbohydrates include energy stores, and so this is glycogen, certainly used as fuel, which are then used for ATP, and as metabolic intermediates, and we'll see that in the few lines down below. So you can see it's much more important than just energy. There's broad interactions across different pathways. So we know that first of all, sugars form the part of RNA and DNA. So we've got ribose and deoxyribose respectively. So ribose is a sugar. And so you can see here that carbohydrates are important in nucleic acids. So it may not be a connection that you would have initially made, but you can see that they're important in that case. We've got polysaccharides, and we'll talk about this in a moment, but essentially poly means many, so many of these units joined together can also have a structural function. Now, when we look at cellulose as an example within the plant cell wall, this gives structure to the cell. So you can see here, it's still a carbohydrate, just that these are linked together as cellulose. And we also know that carbohydrates can be linked to other molecules, in particular proteins, and these are referred to as glycoproteins. So in one of the first lectures, we spoke about proteins going to the endoplasmic reticulum, the Golgi body, for modification. This is one example of that modification process where proteins can have carbohydrates attached, and that is important for their actual function. And it's also true for lipids, so you have glycolipids. And we will see this when we come to talk about lipids in the later lectures. Now, if we move on to terminology, so mono equals 1. So mono means 1, so a single saccharide unit. So as it says here, this is the simplest carbohydrate unit. This is analogous to proteins and the building blocks of proteins being amino acids. But you've got 1 to 20 amino acids, and these are the basic building blocks. So it's the same thing with carbohydrates, but here we're talking about monosaccharides. So these monosaccharides can't be broken down into anything simpler. This is the smallest or simplest unit. Now we're going to start to move into more chemistry now because we are looking at metabolism. But again, don't let this put you off. We will talk about, I will highlight to you really clearly the aspects that you need to know, particularly in this lecture, but more so in the next lecture where we start to look at glycolysis and we go through a lot of the metabolic pathways. But you can see that monosaccharides are just chemicals. So aldehydes or ketones that have 2 or more hydroxyl groups. And so you can see here, dihydroxyacetone has got 1 hydroxyl group and another hydroxyl group here. And again, you can see the carbon-oxygen double bond. Again, you can see the same principle here for glyceraldehyde, and this is the D and the L form. See that here on opposing sides. So these are just examples of simple monosaccharides based on this simple structure. So as a result, they have an empirical formula which is CH2O and then various numbers of those. And you can see that up here, CH2O, CH2O there, and so forth. Now the one that we will talk about the most and the most common is going to be glucose. And we all know about glucose and how important glucose is. And this is the one where we're going to focus from a eukaryotic as well as a prokaryotic level. But this is really the key molecule that is used for energy, but also it is a source of carbon atoms. And that is an important idea, an important distinction to make. These all have lots of carbon, so this is an important source of carbon for these molecules and also then for the host. And I've got some examples here of the most common monosaccharides shown. So we've got D-ribose and D-deoxyribose. So this is for RNA, and this is used in DNA. This is what we've seen. We've seen glucose, D-glucose version of shown here, and then you've got the other ones that are mannose, galactose, and fructose. The galactose, we will see this when we come to talk about the lac operon, and we'll see that galactose is actually converted into glucose by the lac operon in bacteria. But again, you can see these are all very similar. The only difference is really the location of the different groups. As you can see, CH2O, as we saw before, and we've got these various groupings along. So they don't have to be dramatic differences, and this is really quite an important point here. You don't have to have massive differences from one to the other. And also, notice that these are all the D form, not the L form. So again, it shows you that there is preference within the host cell for a particular orientation or an enantiomer of this particular molecule. Now, as we said, the most common form of glucose is the most common monosaccharide, but also how is it presented? It's actually presented or shown in a chair configuration. So we can draw it in this linear portion like this, as a very strung out version, but then it can also then be shown in this particular configuration, that it will actually be bent on itself. And what this black arrow here is showing is that we'll have a linkage taking place, a bond. And so this is going to be an intermediate state, and then we'll go to either of these two states. And so these are both referred to as the chair form, here and here. And so we've got the different types, alpha D glucopyranose or beta D glucopyranose. We'll talk about which one we're interested in, but you can see it's really just the difference between the hydroxyl group in terms of its orientation of alpha versus beta. So indication of alpha and beta refers to the C1 hydroxyl being below or above the plane of the ring. Do you really need to worry about this level of detail? No. But do you need to know that glucose is the important molecule that exists in this chair or open and closed chair configuration? Yes. And certainly we'll go into more detail about the importance of glucose. So, chair form in the beta D glucose version is the most stable. And again, this is more than likely to be due to steric hindrance. And again, here we've got the different configurations. You can see the chair form in this orientation and the boat form here. You can see that with the boat form, it's shown like that there's going to be more interactions here than there is in this chair form. So this is the most stable form, probably from the steric hindrance perspective. Okay, now, carbohydrates as reducing sugars. Now, in the chain form, which is this linear form, glucose can also act as a reducing agent and therefore become oxidized. So remember, this is a redox reaction. So oxidation is loss, reduction is gain. So it can be reduced, so it can gain, and therefore can be, or act as a reducing agent, and therefore can become oxidized by losing the proton. And so in glucose, it's going to be the aldehyde that is oxidized and this becomes a carboxylic acid. And this is important because this reaction allows you to determine the presence of sugars such as glucose in a sample. So whatever the sample is, if you have this reduction potential, it's probably due to the presence of this carbohydrate. So here is the closed form. Here is the linear form, in the chain form. And again, you can see that in the presence of copper, copper 2 plus goes to copper plus, so it's gaining the proton. You end up with this different variation of the molecule. And so this in turn allows for that reaction to take place, and you can also then see that there is a reducing sugar that is present. You can also see that you've got this hydroxide ion, but it's not a full hydroxide ion, it's got a negative charge. This is also going to be reactive as part of this process. Now, at the beginning, we spoke about glycoproteins. Remember that proteins that have been covalently modified. So this is the covalent detection of a carbohydrate unit. Now, this is a precise process, so it's not a random thing. So what this means is that it is a directed linkage. The carbohydrate is placed on a specific residue, specific amino acid, or a specific location within that amino acid, and it is also a regulated process. So that is important. Otherwise, you could have non-specific binding, and that could actually affect the activity of the protein. So where do they attach? They attach to the amide nitrogen atom on the side chain of asparagine. So here we're now being very specific. So it's a specific amino acid. Is asparagine going to be present in most proteins? Yes. If not, then there's obviously no reactivity. But as you can see here, it can also attach to the hydroxyl atom on the side chain of serine and threonine. So the likelihood is that all proteins will have at least one of these individual amino acids. And as you can see, about 50% of proteins exist in this configuration. So they are modified. That's a lot. 50% of all proteins have some further modification. So that is important for the activity of that particular molecule. So that brings us really to the next point in terms of these monosaccharide modifications. Well, why? Why is this occurring? So you have these reactions between alcohols. You have them between amine groups and phosphates. And what this does is by having these additional groups being added, it increases the diversity and also the versatility of the protein. Because what you can have is no modification, one or multiple modifications. And as a result, that is going to affect the activity of that particular protein. You could also imagine that that might be segregated in the sense that a protein in one organ might not be modified, the same protein in a different organ might undergo some covalent modification, and that then dictates or alters its function in a different organ or in a different cell. This is also linked in with signaling and facilitating the metabolic fate of sugar, so determining the pathways of metabolism. Now, the signaling idea is the transmission of information. And so what this is referring to is external signals, external stimuli to the inside of the cell. And so cells have to be able to respond to the environment. Signal transduction is part of that. We'll devote an entire 2 lectures to signal transduction, and there we will see that proteins are key molecules in the signal transduction pathway, and they can also be modified through this particular mechanism. So I'll just stop here. So as you can see, you've got the different terms or types of sugar. So you've got fructose here, and acetyl galactose amine. So again, you can see that you've got an amino group here with a different version of glucosamine, etc. You would have heard of glucosamine, particularly on TV, for people that are suffering from joint problems. As an example, you've got all of these other groups. You've got sialic acid, which is something that's very common within the body. You've got this molecule here, glucose 6-phosphate and dihydroxyacetone phosphate. These two do highlight them, because we will talk about these in subsequent lectures, and they will be very important, particularly glucose. You can see here that the glucose can be phosphorylated. It's got a phosphate group attached. These are very important. And again, here we've got another monosaccharide with a CH2O grouping. So very important groups. Now, I'm just going to stop there. This is a relatively short lecture, but I'll stop here. We'll come back in the next lecture and start to talk about the bonding and structure of glucose. Okay, welcome back. So we finished off by talking about the different types of modification. And now let's move on to talk about monosaccharides and the linkage. And so when we link monosaccharides together, we end up with disaccharides. So di equals 2 and poly equals more than 2 together. And so what this allows you to do is to generate complex carbohydrates. Now, if we start off with the simplest version, a disaccharide, as it says here, 2 sugars, which is what I said just up here. These are linked via the O-glycosidic bond. So glycosidic refers to glucose. This is the nature of the bond. So just as we've seen with amino acids having a peptide or an amide bond, a glycosidic bond is for glucose or carbohydrates. So some sugars that we would have come across before is sucrose, is very common. Lactose, we know people that have a lactose sensitivity or an issue with ingesting lactose milk, and maltose. These are the most common types of disaccharides that we will see. These can be hydrolyzed, and so what this is referring to is water breaking them apart, hence a hydrolysis reaction, and it occurs by the enzyme sucrase, lactase, and maltase, respectively. And remember, ASE tells you that these are enzymes. Whenever you see ASE, so lactase, sucrase, maltase, they are enzymes, and so they have the activity. The first part of the name is telling you the substrates of sucrose, lactose, and maltose. So easy way to remember and also to learn about what is actually the target. Now, as I mentioned, polysaccharides are multiple sugar units that are joined together, but these are joined together again in sequence. And when we look at these, the pattern can vary depending on the bond that occurs. But what we are interested in is this molecule here, glycogen. This is an important form of storage of glucose in humans. Those are eukaryotic cells. This is the one that we are going to be talking about a lot, so get very familiar with glycogen. The alternative, if we were to talk about plants, would be starch. So glucose can be stored as starch, and we know that things like potatoes are rich in starch, they're therefore rich in glucose. Now, we're not potatoes, we're not plants, so we'll talk less about this. So much of what we are going to focus on, particularly in the metabolic pathways, is going to be around the glycogen idea. Okay. Now, you can see here that we have different types of bonds. We have an alpha 1,4 glycosidic bond. Now, what we have here is one monosaccharide, so one in the middle, and here's the second monosaccharide. The alpha 1,4 is telling you the bond. So this is the 1 position. So we start from this location, and we must be going that way. How do we know? Because this is the 4 position. Okay, and we're going left to right. So this is the 1 to the 4 position, and we have this bond. So this is the glycosidic bond that we see here. This can be repeating, just like we've seen. This is now able to form another glycosidic bond, and it can keep going. You also have the option of an alpha 1,6 glycosidic bond. So we have the 1,4, but we've also now got this bond occurring up here, and again between the 1 and the 6 position. So that's going to affect this hydroxyl group here, and this gives you now an ability to branch of the monosaccharides. That's important because that's part of the storage. So these are the 2 types of bonds that we will encounter. You need to know these 2 types of bonds and the structure of this interaction. But again, it's fairly simple and straightforward. So what does a disaccharide look like? So again, alpha, the D, so the alpha is the location of the bond. The D is telling you the enantiomer, glucosyl pyranosol. So it's glucose. We've got the beta, D, fructofuranose. So this is going to be fructose. You can see the different structure. So again, you've got here galactose and glucose. You've got glucose and another glucose. And what do you see? These together give rise to sucrose, lactose, and maltose. So the different combinations of individual sugars give rise to these different components. So small differences, but you can see that you can have quite a dramatic diversity between the 2. So again, 1,4 bond that is occurring, and again, same thing here. Here we've actually got a slightly different version, but you can see the same types of interactions. Okay, so a fairly short lecture, which is fine, because I think we really want to just focus on those key issues relating to carbohydrates. The next lecture is going to get quite detailed because we're going to go into like version of the lecture, which I think is